Hello, everyone. My name is Vijay, second year grad student in the GGR department with Dr. Amanda Bajwa. And today I'm very much pleased to host and introduce Dr. Eric Greer to our UTSSC GGI virtual seminar series. Dr. Greer is an assistant professor in pediatrics at Harvard Medical School, as well as a PI at Boston Children's Hospital in the Division of Newborn Medicine. And Dr. Greer received his PhD from Stanford University under the mentorship of Dr. Anne Brunette. He continued his training with a postdoctoral position in the Brunette lab, followed by a second postdoctoral fellowship at Harvard Medical School under Dr. Yancey. And since his last position, he has stayed at Harvard, where he has mentored students and instructed classes in various qualitative and quantitative analysis. And he has also received many honors, including awards and grants from NIH, the Broad Institute, the NSF, and most recently, the Presidential Early Career Award for Scientists and Engineers. And he is a reviewer for major publications, including Cell, Nature, and Science, and serves on the editorial board for Environmental Epigenetics and Epigenomes. And he has given over 67 talks and presentations, and currently has over 30 peer-reviewed publications, with more than half of those being his first authored. And I'm thankful to Dr. Greer for accepting my invitation for our seminar series, as well as making time to chat with us today regarding his research and his science. So with that, I'd like to hand off the presentation to Dr. Greer. Thank you.
Great. Thank you very much for that introduction. And before I begin, I'd just like to also thank you and the students for inviting me. It's always a really special honor, I think, to be invited by the students. So today I'm going to tell you guys about two different vignettes from my lab, both of which have been recently published, one of which was published this week, on examining the role of epigenetics in the evolution of multicellularity and our identification of a novel RNA methylation and how that regulates stress resistance. But before I begin, I thought I would give you a broad overview of what my lab does. We're really interested in the molecular mechanisms of epigenetics, and we study this across the entire spectrum of the definition of this word. And people use this very loosely and very stringently, and we really study it all, anything which falls under the umbrella of epigenetics. So at the most stringent end of this spectrum, we study transgenerational epigenetic inheritance. So this is a non-genetic inheritance of information from ancestors to their descendants, which happens in multiple generations in a non-Mendelian, non-genetic manner to regulate complex phenotypes. We primarily use the nematode C. elegans to study these phenotypes because it has a very short generation time, and I and others have identified very robust transgenerational inheritance phenotypes. So we try and identify molecularly and mechanistically what specifically is being passed from ancestors to their descendants to regulate these complex phenotypes. On the next layer down, we study imprinting, which is non-genetic information being passed from parents to their children and no further. And this work we primarily do in human cell lines and in mice to try and identify how imprinting is established, how it's maintained, and potentially how to reverse it to rescue some of these very debilitating imprinting disorders which happen in children. A portion of my lab is focused on identifying what the cellular consequences of chromatin modifications are. And this is work we do in C. elegans, Chlamydomonas, humans, mice. Recently, we've started examining viruses and how viruses will hijack the cellular machinery, the cellular epigenetic machinery, to evade the host immune system. A portion of my lab is focused on identifying and characterizing novel epigenetic modifications, as well as the enzymes that add, remove, and recognize those modifications, and then examining what their role is in a variety of different biological processes. And this is work that we do in humans, mice, and C. elegans. And finally, a portion of my lab is trying to repurpose Dictionalium Dyscodium to develop it as an epigenetic model organism that we think will be useful for addressing all of these questions of epigenetics. But initially, we've been using it to examine the role of epigenetics in the evolution of multicellularity. So today, as I said, I'll be telling you two different vignettes, one on our identification of a ribosomal RNA methyltransferase and how that regulates stress resistance, and the second one on examining the role of epigenetics in the evolution of multicellularity. So expressing the appropriate repertoire of proteins in response to various environmental cues is an essential and environmentally and evolutionarily conserved process. Now, historically, this was thought to be regulated solely by the transcriptome and the genome, and that we had a homogeneous ribosome which would translate whatever transcript it was presented with. But more recently, it's been demonstrated that there are actually unique ribosomes, and these can translate specific sets of transcripts. So this has led to this theory of ribosome heterogeneity. So the ribosome consists of 4 ribosomal RNAs, the 28S, the 18S, the 5.8, and 5S, as well as roughly 80 different ribosomal proteins. So work actually in the 1980s by Ramagopal and Ennis in Dictionalium demonstrated that under different cellular states, the ribosome consisted of different ribosomal proteins. So this led to this theory about ribosome heterogeneity, that this could be an additional layer of regulation of which proteins get translated. To support this theory, work in yeast, C. elegans, and mice has demonstrated that under different stress conditions, the ribosome is bound to a unique set of stress resistance transcripts to allow for the translation of stress resistance proteins. And additionally, some very elegant work from Maria Barner's lab demonstrated that they immunoprecipitate different components of the ribosome, and they found that they would be bound to a unique set of transcripts. So together, this work has over the past decade or so, has really lent a lot of strength to this theory that different ribosomal proteins make up the ribosome, and this can help to specify unique sets of ribosomes, special ribosomes, which will translate special transcripts. So we hypothesized that potentially other modifications to the ribosomal RNAs could also contribute to this ribosome heterogeneity. So to test this, Zach and Andrew decided to perform a directed RNAi screen in the nematode C. elegans, where they would knock down all putative RNA methyltransferases, extract the total RNA, and then perform high-performance liquid chromatography coupled with mass spectrometry, which is a technique which allows us to quantify all the different types of modifications that occur on RNA. Under the assumption that roughly 80 to 90% of total RNA is ribosomal RNA, so if we were to see a significant change in total RNA methylation, it would presumably be due to changes in ribosomal RNA methylation. So Zach and Andrew performed this screening, and they identified one transcript, C38D4.9, which when they knocked it down, caused a roughly 40% decrease in N6 methylation of adenosines. So M6A, methylation on the N6 position of adenosines. So this suggested that C38D4.9 could potentially be an M6A methyltransferase. However, we were aware that potentially this phenotype could be due to off-target effects of RNAi. So to see if these changes in M6A was really due to C38D4.9, we obtained 2 different genetic null mutants of this gene, which we renamed METL5 due to its homology to the mammalian proteins. We extracted the total RNA and again performed HPLC coupled with mass spectrometry. And what we found was that deleting this gene caused roughly a 50% decrease in M6A levels and without changes to other modifications, suggesting again that it could potentially in vivo be an M6A methyltransferase. To identify which substrate would be modified of the RNA, Zach and Andrew ran the RNA out on a gel and then extracted the 28S, the 18S, and the 5.8 and 5S, and performed HPLC on each of those individual substrates. And what they found was that in response to METL5 deletion, there was no change in mRNA methylation or methylation on the 28S, 5.8, or 5S. However, there was almost a complete elimination of methylation of the 18S ribosomal RNA. Now, the 18S has been shown in humans and in zebrafish to be methylated, to be M6A methylated at a single residue, adenosine 1832. And we want to identify whether this residue was being methylated in C. elegans and whether it was being methylated by metal 5. So C. elegans do have this residue being conserved. It's adenosine 1717 in C. elegans. So to examine whether it was methylated and being methylated by metal 5, Kostis performed directed RNA cleavage followed by p32 labeling and thin layer chromatography, which allows us to identify whether a single residue is methylated or not. And so here, just for examples, you can see that when you run differently methylated residues out on a TLC, adenosine runs very differently from M6A. So Kostis performed this examination looking at adenosine 1717 in wild-type worms and in the metal 5 mutant worms. And what he found was that indeed in C. elegans, this residue was methylated, roughly 98% of the time. However, in the metal 5 mutant worms, this residue was almost completely unmethylated. So suggesting that metal 5 could be 18S methyltransferase on adenosine 1717. To test to see whether metal 5 was directly methylating this residue, we GST tagged and bacterially purified this protein, and then incubated it with 18S, which we had purified from metal 5 mutant worms, so it was devoid of methylation at this particular residue, and used deuterated methyl SAM as our methyl donor, and then performed high-performance liquid chromatography coupled with mass spectrometry to try and quantify changes in methylation. And what Andrew found was that metal 5 was very efficient at methylating 18S, which we purified from metal 5 mutant worms, but was unable to methylate 18S, which we purified from the wild-type worms, which already had that residue basically constitutively methylated. So this suggests that metal 5 really is an 18S methyltransferase. However, we wanted to test to see if this activity was really due to metal 5 itself rather than spurious proteins, which we were potentially purifying from the bacteria. So Andrew mutated the putative catalytic domain of metal 5, this NPPF domain, to an APPA, and again performed these in vitro methylation assays. And what he found was that this catalytically dead version of metal 5 was unable to methylate 18S purified from metal 5 mutant worms, suggesting that in vitro, metal 5 really was an 18S methyltransferase. Next, we wanted to determine whether metal 5 was specifically methylating adenosine 1717. So we synthesized oligos which contain adenosine 1717 as well as the flanking nucleosides, and again performed our in vitro methylation assays. And what we found was that metal 5 could methylate these oligos very efficiently, whereas again the catalytically dead version of the enzyme was unable to methylate them. To determine metal 5's specificity for this particular adenosine 1717, we mutated that adenosine to guanosine, and we found that that completely eliminated the capacity of this enzyme to methylate these oligos, despite the fact that there were other adenosines present in this oligo, suggesting that metal 5 does have a certain degree of sequence specificity towards adenosine 1717. So together, these results had suggested that metal 5, both in vitro and in vivo, was an 18S ribosomal RNA methyltransferase, and it was methylating this specific residue, adenosine 1717. Now we wanted to identify what the functional consequences of methylating this particular residue were. And if you recall from my introduction, I had mentioned how people had identified that ribosome heterogeneity played a role in responding appropriately to various stresses. And so what we did was we exposed the metal 5 mutant worms to a battery of different stress assays and measured their survival. And what we found was that the metal 5 mutant worms showed increased resistance. Here I'm just showing you heat stress resistance, but they also showed increased resistance to UV stress, oxidative stress, and cold stress. And if we take their survival out for the entirety of their lifespan, you can see in this Kaplan-Meier survival curve that the metal 5 mutant worms survive about 150% longer than their wild-type counterparts. So this is suggesting that this enzyme, which is methylating the 18S at this particular residue, is conferring altered stress resistance. We want to determine whether this increased stress resistance was really due to metal 5 and its catalytic activity. So we created transgenic rescue strains where in the METL5 background, we overexpressed either the wild type form of METL5 or this catalytically dead version of METL5. And here you can see that when we looked at 18S methylation, the wild type version reverted the 18S methylation back to the wild type levels, but the catalytically dead version was unable to do that. And when we examined the stress resistance of these worms, what we found was that, as before, the METL5 mutant worms showed increased stress resistance relative to the wild type worms, but the rescue with the wild type version of METL5 completely reverted the stress resistance back down to wild type levels, whereas the catalytically dead version was unable to do so, suggesting that this increased stress resistance was really due to METL5 and its capacity to methylate the 18S at adenosine 1717. So we've identified this enzyme, which is methylating the ribosome, and it's causing this alteration in stress response. Is this due to changes in translation? To assess this, we decided to administer a broad translation inhibitor, cyclohexamide. And what we found was that, as before, the METL5 mutant worms show increased stress resistance relative to the wild type worms. However, when we inhibit translation, the METL5 mutant worms no longer display this increased heat stress resistance. We not only observed this for heat stress, we also saw this in response to UV stress as well. So we've identified this 18S ribosomal RNA methyltransferase, which when it's deleted, is conferring extended stress resistance because of some changes in translation. Next, we were interested as to what the changes in translation were that were being regulated by this methylation of the specific residue. So we performed S35 methionine labeling experiments where we fed wild type or METL5 mutant worms radioactive methionine, and we either left them unstressed or stressed them. And then we measured either by scintillation counting or by running out on a gel to see if there was any change in the amount or specific proteins which were being produced. And actually, to our surprise, what we found was that there wasn't really a global change in translation in response to deletion of this 18S methyltransferase, despite the fact that it's having this pretty dramatic effect on stress resistance. And similarly, while 37-degree heat shock does have a dramatic effect on which proteins are being produced, this was METL5 independent. So at least by these global measurements of translation, we didn't really see any differences between the METL5 mutant worm and the wild type worms. So this was a bit perplexing, but we thought, oh, well, maybe it's because there's a change in specific translation that we aren't really able to assess by these global measurements of translation. So to examine this, we decided to perform ribosome sequencing under the same conditions at 20 degrees or at 37 degrees in the wild type worms or the METL5 mutant worms. And again, as you can see by this heat shot, this heat map, we recapitulated what we found by these global assessments. If you look at this from a global perspective, there wasn't really any difference between the wild type worms and the METL5 mutant worms. However, there was a single gene which reached above our level of statistical significance, CYP29E3, which did appear to be statistically significantly differently bound by the ribosome when we have or when we don't have this methylation of the 18S. So this is always a little bit worrisome when you do a genome-wide analysis and you get a single transcript. But nonetheless, we wanted to test this to see if this was a real result and if CYP29E3 could be responsible for the altered stress resistance that we were observing in the METL5 mutant worms. So what Noah first did was she performed real-time RT-PCR to see if levels of the transcript of CYP29E3 changed in the METL5 background. We found that they didn't. Although CYP29E3 transcript levels increased in response to heat shock, they increased to the same amount in the wild type and the METL5 mutant background. Noah also performed polyzone profiling followed by RT-PCR, and what she found was that she was able to validate the results of the ribosome sequencing, that there was indeed an increased binding of the ribosome to this one transcript, CYP29E3, under these heat stress conditions, which we didn't observe in the METL5 mutant worms. So our ribosome sequencing data, at least for this gene, was validated. And next we wanted to see if CYP29A3 was important for these stress resistance phenotypes, which we observed. So first we had to ask, what is CYP29A3? Well, Kulisol had previously demonstrated that this is an enzyme which oxidizes omega-3 and omega-6 polyunsaturated fatty acids to produce these signaling lipids termed eicosanoids. So eicosanoids play a role in, primarily they've been identified in regulating inflammation, but they also play a role in regulating a variety of other phenotypes. And they exist as these lipids which will signal between cells and between organs. So we wanted to identify whether CYP29A3 and the eicosanoids, which it was supposed to be producing, were responsible for the altered stress resistance of the metal-5 mutant worms. So to examine this, we first obtained a genetic null mutant of CYP29A3, and we found that this mutant phenocopied the metal-5 mutant worms. So when we exposed it to heat stress, it also showed increased heat stress relative to the wild-type worms. So next, we want to identify whether CYP29A3 was functioning in an independent parallel signaling pathway to metal-5, or if it was functioning in the same signaling pathway. So we performed epistasis experiments. And what we did was we overexpressed CYP29A3 in the context of the metal-5 mutant background. So as before, we found that metal-5 mutant worms show increased stress resistance relative to wild-type worms. However, if we overexpress this one transcript, this CYP29A3, this was sufficient to reverse the stress resistance back down to the wild-type levels, suggesting that CYP29A3 is functioning downstream of metal-5 to regulate stress resistance. So is this due to CYP29A3's function on these polyunsaturated fatty acids? First, we wanted to measure to see if the metal-5 mutant worms had altered levels of these eicosanoids. So we performed gas chromatography coupled with mass spectrometry. And what we found was that the metal-5 mutant worms, while they had similar levels to wild-type worms, shown here in orange, as the wild-type worms of the immediate precursors, these omega-3 and omega-6 polyunsaturated fatty acids, they did display reduced levels of these specific oxidized eicosanoids, which CYP29A3 is responsible for producing. So are these eicosanoids themselves responsible for the increased stress resistance that we see in the metal-5 mutant worms? To assess this, we performed refeeding experiments. And here I'm just initially showing you refeeding in the CYP29A3 background. So what we did was we fed the worms either the precursors, these omega-3 or omega-6 polyunsaturated fatty acids, or the oxidized substrates, these eicosanoids, which they're producing. Oops. What we found was that when we fed the omega-3 or omega-6 polyunsaturated fatty acids, they had no effect on the phenotype. The CYP29A3 mutant worms still displayed this increased heat stress resistance. However, when we fed any of these downstream eicosanoids, they were sufficient to reverse the stress resistance back to the wild-type levels. And we saw the same thing in the metal-5 background as well. So just refeeding them with these particular eicosanoids, which CYP29A3 is responsible for producing, was sufficient to reverse the stress resistance back down to wild-type levels. So what we think that we've identified is that metal-5 is methylating the 18S at this particular residue, adenosine 1717, and this is causing altered binding of the ribosome to this transcript CYP29A3, and therefore presumably reduced production of the protein cytochrome P450 enzyme, and it can therefore no longer oxidize these omega-3 and omega-6 polyunsaturated fatty acids to produce these signaling lipids, these eicosanoids, and that causes this altered stress resistance. So this is quite amazing that we identified this enzyme which is able to methylate a single residue on the 18S, and this is able to have these really dramatic consequences in terms of stress resistance and production of lipids. So we're pretty excited about this project, and there are a number of next step questions which we're currently trying to pursue. We're very interested in mutating this specific adenosine on the 18S to examine whether that alone is sufficient to recapitulate these phenotypes. We also want to examine where throughout the worm these enzymes and these lipids are functioning, where the lipids being produced, where are they signaling to. We're also interested as to whether this 18S methylation is affecting the composition of the ribosome, the proteins which are making up the ribosome, or if it's somehow just specifically affecting the ribosome's capacity to bind to CYP29A3 and potentially other transcripts. And more broadly, we're also interested in other instances of ribosomal RNA methylation to examine whether they affect ribosome heterogeneity and other processes within the organism. Okay, so now I'm going to switch gears pretty dramatically and tell you about this other story, examining epigenetics in the role of the evolution of multicellularity. So unicellular organisms are relatively diverse in terms of their appearance, shape, morphology, where they survive. But there was a real explosion of diversity upon the transition to multicellularity, which allowed organisms to adopt very dramatically different phenotypes, survive in different environments, and live much longer. And obviously, this also led to our own evolution, for better or for worse. However, how the transition from unicellular to multicellular organisms happened is still an unanswered question. Mostly theories about this have led to, are around the idea that there was the acquisition of new genes or new transcription factor networks, which allow this transition to multicellularity. However, when we examine ancient multicellular organisms and compare them to unicellular organisms, oftentimes they have the same genes and the same transcription factors, suggesting that there could be other alternative factors which could have helped to contribute to this jump to multicellularity. And we postulated that epigenetics could have been one of the critical factors which facilitated the transition to multicellularity. And part of the reason why we postulated this was that at this transition time, there was a real explosion of epigenetic diversity. Histone demethylases evolved around this time, and a lot of the chromatin modifying enzymes duplicated and became much more dynamic. So we postulated that epigenetics could have been critical for this transition to multicellularity. So for multicellularity to have evolved, several critical traits had to be acquired. Cells had to learn how to adhere to each other, to communicate with each other. Cellular specialization occurred around this time, whether it's cause or consequence is still unclear. And also cells had to learn how to be altruistic, where they would, in the most extreme case, where they will kill themselves for the survival of the other cells in the organism as a whole. So some of these traits are clearly due to the acquisition of new genes, adhesion, cohesion molecules, for instance, but some of them are still incompletely understood. And again, we speculated that potentially epigenetics could be important for these processes. So to examine this, we turned to this really cool model organism, Dictionalium Dyscodium, which we think is really ideally situated to address this question. So the reason why we think this is because this is one of the rare organisms which is both unicellular and multicellular and transits regularly back and forth between these two states. So what I mean by that is that normally Dictionalium is a single cell amoeba, but when it consumes all of its food, it will send out this chemical signal, and this causes it to aggregate into a multicellular organism, initially forming a multicellular mound, and then it will transition to become a slug, and it will go searching for food. And if it can't find any food, it will transition again. It will plop itself down, it'll form a stalk and spores, and then eventually it will germinate and send those unicellular spores to foreign locations to find food. So this is a really dramatic and dynamic process. This all occurs within 24 hours. And it's clearly epigenetic because the organism has the exact same genome at its unicellular and multicellular stages. So something non-genetic has to be contributing to this property. So we thought it was kind of ideally situated to study whether epigenetics was really involved in the evolution of multicellularity, to do not only the observational science, but also to be able to manipulate the genome and the epigenome to test our theories as to whether epigenetics was important for multicellularity. So part of the reasons why Dictionalium are great is because it has a very small genome, so it's relatively affordable to do lots of epigenomic analysis. It's very easy to culture in the lab to manipulate both environmentally and genetically. As I said, it's both unicellular and multicellular. And for some other questions which we're interested now in addressing, in addition to these, is it has these very clear instances of both selfish and altruistic cells. So what I mean by that is normally, so here during this fruiting body stage, about 20% of the organism exists in the stalk, And these cells will kill themselves for the survival of the other 80% of the cells. But just 20 hours earlier, those cells were only concerned with their own survival. So how do they make that transition to becoming altruistic? And is epigenetics involved in that? So that's not the question which we're initially addressing, but it's something which we're excited about addressing in the future. So we initially wanted to determine whether epigenetics and chromatin modifications were important for this transition to multicellularity in Dictyostelium. So to see whether this theory had any weight, we first wanted to to test this in a relatively facile way, where we administered various chemicals which affect chromatin modifications and measured whether or not they affected the timing that it took for Dictyostelium to become a multicellular organism. So to assess this, we fed trichostatin A, which is a histone acetylation inhibitor, as well as sinfungin and Bix 01294, which are methylation inhibitors. And then we measured the timing that it took for the Dictyostelium to become multicellular. And what we found was that when we administered all three of these drugs, it did indeed slow the timing that the Dictyostelium took to proceed to the multicellular stage, as well as slowed their entire developmental progression. So this would suggest that potentially chromatin modifications could be playing an important role in the transition to multicellularity in Dictyostelium. However, we were also very well aware that potentially this could just be making the Dictyosteliums, these chemicals could just be making the Dictyostelium sick, and we could basically be slowing all different processes which they were involved in. So first we wanted to test that before we went further with this project. And to test this, we initially performed chemotaxis assays. So the Dictyostelium can move towards a chemical gradient exceptionally well. So we measured whether administering these chemicals which slow multicellularity also affected other properties. What we found was that while TSA did significantly decrease the rate of chemotaxis, neither sinfungin nor Bix significantly affected chemotaxis capacity. Additionally, we measured their effects on apoptosis. And again, what we found was that while TSA did have a significant effect on apoptosis, neither sinfungin nor Bix did significantly affect them. So these are suggesting that at least for these histone methylation inhibitors, that they could be really quite relatively specific for this phenotype of multicellularity without generally making the Dictyostelium sick. So this gave us enough confidence to examine the epigenome in more detail in this organism. So what we did was we performed epigenomic profiling at the unicellular stage, the vegetative stage, at the streaming phase when the Dictyostelium are just beginning to become multicellular, at the mound stage when they truly are multicellular, and the fruiting body stage, which is their final developmental progression. And what we did at these 4 different developmental stages is we performed RNA sequencing, chromatin immunoprecipitation, followed by sequencing for 7 different chromatin modifications, and attack sequencing, which is this method of globally examining which regions of the chromatin are open and accessible for transcription and which regions are closed and quiescent. So what we found was that when we performed attack sequencing, this method of globally examining chromatin accessibility, when we examined the unicellular stage relative to the various multicellular stages in this metagene analysis where we're condensing all of the data into a representative gene, we found that there were indeed changes in the chromatin accessibility pattern. So the vegetative, the unicellular stage, was more open and accessible immediately following the transcriptional start site, and this decreased as the organism progressed into the more multicellular stages, whereas there's more chromatin accessibility in the gene body in the multicellular stages. And this dramatic shift in chromatin accessibility can be visualized when we analyze this in a heat map format, where you see that the unicellular stage clusters very separately from the various multicellular stages. And similarly, if we condense all of this genome-wide data into an individual point and perform a principal component analysis for the ease of comparison, what we see is that the unicellular stage clusters very separately from the various multicellular stages. So this is suggesting that at this global chromatin architecture change, there are really dramatic changes when the dictyostelium is in its unicellular stage or in these various multicellular stages. We next want to examine more specifically whether there were other more precise chromatin modification changes which were occurring as the dictyostelium was transitioning. And so Liz Polina performed ChIP-seq chromatin immunoprecipitation followed by sequencing for 7 different chromatin modifications. And what she found was that, again, when she performed this principal component analysis, condensing all of this genome-wide data into one point for the ease of comparison, what she found was that these 3 typically activating chromatin marks, which are associated with chromatin activation, H3K4 trimethylation, H3K27 acetylation, and H3K4 monomethylation, allowed the unicellular state to be clustered, to be significantly distinguished from the various multicellular stages. Whereas other chromatin modifications were less good at this. So H3K36 trimethylation, H3K27 trimethylation, H3K9 trimethylation, and histone H3 itself. So this would suggest that there might be more dynamic chromatin modifications, such as the activating chromatin modifications, and a separate set of more housekeeping type chromatin modifications, which are maybe more constant as dictyostelium transition between different developmental stages. We also wanted to examine how the transcriptome was changing as dictyostelium transitions from this unicellular to various multicellular stages. So we performed RNA sequencing. And here we were able to basically recapitulate what others had previously shown by microarray analysis, was that, as you would anticipate, there are a lot of genes which decrease as dictyostelium transition to multicellularity, some which stay constant, some which go up and then down, some which just go up. And if we, again, perform a principal component analysis, we find that the unicellular stage, shown here in blue, clusters very separately from the various multicellular stages. So as I said, this kind of recapitulated what others had previously shown by microarray analysis, but now we had these data sets which were matched. So they were all done at the same time, so we could examine how these epigenetic changes were affecting transcriptional changes. So Simon initially wanted to examine the transcriptome to see how gene expression was changing upon unicellular to multicellular transition. And what he found when he did a gene ontology analysis was that the genes which were particularly enriched at the unicellular stage were genes which are important for basic biological processes, kind of as you would anticipate, genes involved in translation, protein synthesis, metabolism. And the genes which were enriched on the multicellular stage were, again, genes which you would anticipate as being essential for multicellularity. So genes involved in development, differentiation, cell-cell communication, adhesion. Excuse me. So again, this was kind of recapitulating what we would anticipate in terms of the transcriptional changes. But this allowed us to examine whether these epigenetic changes or these chromatin modification changes were affecting transcription and helping to drive multicellularity. So first we want to examine whether the chromatin modifications were behaving the same in dictyostelium as they've been reported in other eukaryotes. So first what Simon did was he examined the 1,000 most highly expressed genes and the 1,000 most lowly expressed genes at each different developmental stage and compared that to chromatin accessibility. And what he found was that genes where the chromatin was open and accessible for transcription were indeed more highly transcribed, and those which were more closed and quiescent were more lowly transcribed at each of the different developmental stages. Additionally, if we zoom in on a representative gene, in this case, a transcription factor, SRFA, what Simon found was that the chromatin modifications behaved in dictyostelium pretty much the same way that they had been reported to behave in other organisms. So SRFA is a gene which turns on upon multicellularity. And what we see is that there's an increase in chromatin accessibility in the promoter of this gene when the dictyostelium transitioned from unicellular to the various multicellular stages. Additionally, this is accompanied by an increase in H3K4 trimethylation, this chromatin activating mark. as well as a decrease in this repressive mark, H3K27 trimethylation. So suggesting that chromatin modifications in Dictyostelium behave pretty much the same way that they do in other eukaryotes. So now we have this comprehensive epigenomic data set at the unicellular and various multicellular stages, and we want to see if we could use this to try and identify things which were important for multicellularity. So if you recall from the intro, the main things which have been identified thus far to regulate multicellularity was the acquisition of new transcription factors and new genes. So we wanted to examine whether changes in chromatin modifications would affect transcription factor binding sites. So would specific transcription factor binding sites become accessible and open for transcription upon the transition to multicellularity? So Simon searched for motifs which became accessible upon multicellularity, and he identified this MADS box transcription factor was enriched in the multicellular stages. So SRFA, this gene which I just mentioned, is actually a MADS box transcription factor which increases dramatically upon the transition to multicellularity. So we want to test to see if SRFA was therefore important for multicellularity in Dictyostelium. So to test this, we obtained a genetic null mutant of SRFA, and we measured the time that it took this mutant to achieve multicellularity. And what we found was that SRFA deletion did indeed slow the timing of multicellularity. However, like the exact same way that we tested the chemicals that we tested to see if they affected other properties of the Dictyostelium, we also wanted to see if SRFA was really specific for multicellularity or if it was just generally making the Dictyostelium sick. So what we found was that when we deleted SRFA, it also decreased this organism's capacity to chemotax, as well as decreased its proliferation rate, suggesting that SRFA, while it might be affecting multicellularity, it was also affecting these other properties as well. So we wanted to see if we could use the epigenomic data to identify things which would maybe be more specific for multicellularity rather than just generally making the Dictyostelium unhappy. So to examine this, what we decided to do was compare the epigenome of Dictyostelium to other eukaryotes, which had also been epigenomically profiled. And so we compared the epigenome to C. elegans, a multicellular eukaryote, and S. pombe, a unicellular eukaryote, both of which had been epigenomically profiled. So first, Simon identified these 2,500 genes, which were well conserved across all three species. We could identify the clear homologs. And as you would anticipate, these 2,500 really well conserved genes are important genes. They're all more expressed in each of those species. And if you do a gene ontology analysis of these 2,500 genes, they mark basically all the essential evolutionarily conserved processes. So could these 2,500 genes be important for regulating multicellularity? So first Simon looked at gene expression of these 2,500 genes in the unicellular and multicellular Dictyostelium and performed a principal component analysis with C. elegans and S. pombe. And what he found was that by the third principal component, the unicellular Dictyostelium clustered more closely to S. pombe than it did to the multicellular Dictyostelium, suggesting that these 2,500 genes might indeed be important for multicellularity. So what Simon wanted to identify was genes which were open and accessible and transcriptionally active in the unicellular dicty and the S. pombe, but closed and quiescent in the C. elegans and the multicellular dicty, and vice versa, genes which were open and accessible for transcription and expressed in the multicellular dicty and C. elegans, and closed and quiescent in the unicellular states and organisms. So he performed this analysis and he identified 67 genes which were open and expressed in the unicellular states and 52 genes which were open and expressed in the multicellular states. So next we want to determine whether these multicellular genes, these 52 genes, were enriched in all the cells at once, or if they were enriched in specific subpopulations of the cells. So to examine this, we set up a collaboration with Yihao Wang, a graduate student in Paul Greer's lab, and we performed single-cell RNA sequencing. And what we found was that, as you would anticipate, through this clustering analysis, that the unicellular stage clustered very separately from the various multicellular stages. And additionally, the single-cell RNA-seq could recapitulate what we found by the bulk RNA-seq of these genes, of the unicellular enriched genes. We were able to show that, yes, indeed, they showed higher expression in the unicellular stage in the individual cells than they did in the multicellular stage, and vice versa. The multicellular genes showed higher expression in the multicellular stage. And just to give you a few examples of these genes and their expression, we see that SRFA, this transcription factor, which I mentioned earlier, it is indeed enriched, shows enriched expression in the multicellular stage, as does this putative histone deacetylase, HDAD, AMMR1, and this SMC-like protein, SMCL1. So through this epigenomic analysis, we were able to identify conserved genes whose epigenome was changing in multicellular states and multicellular organisms. And next, we wanted to test whether these genes, which we had identified bioinformatically, were actually important for multicellularity. So because Dictionalium is a genetically amenable organism, we were able to actually test what we had identified bioinformatically. So Simon proceeded to knock out a bunch of these genes and then test their effects on multicellularity. And what he found was that when he knocked out 3 of these additional multicellular genes, in addition to SRFA, that each of them slowed the timing, or in the case of SMCL, completely inhibited multicellularity from happening. Whereas when he knocked out a unicellular gene, HSPH2, this did not affect the transition time of multicellularity. So this was really exciting. We had identified genes bioinformatically based on their epigenetic signature, which we thought would be important for multicellularity. And Simon was able to test them and show by knocking them out that they were indeed necessary for multicellularity. However, again, we wanted to test to see if these genes were really specific for multicellularity, or if they were, again, like SRFA, simply making the Dictionalium a little bit sick. And what Simon found was that knocking out none of these other multicellular genes had any effect on the capacity of Dictionalium to chemotax, whereas when he knocked out this unicellular gene, HSPH2, that did slow the chemotaxis capacity of Dictionalium. Additionally, knocking out these genes didn't affect their apoptotic rate or their response to an extreme heat shock as well. So this suggests that these genes, which we find were necessary for multicellularity, are relatively specific for multicellularity. So next, Simon wanted to test to see if any of these genes that we had identified as being necessary were also sufficient for multicellularity. So he decided to overexpress them. And here I'm showing you the overexpression of SMCL1, this SMC-like protein. And what he found was that, actually a little bit surprisingly, was that when he overexpressed SMCL1, it also seemed to slow multicellularity. So this gene, which when he knocked it out, completely stopped multicellularity from happening. When he overexpressed it, it accelerated multicellularity, suggesting that we really need appropriate levels of expression of this one gene. However, he did observe this other phenotype, which was quite obvious when he just looked at the Dictionalium, where he overexpressed SMCL1, which was that the organisms, the multicellular organisms, appeared much, much larger than the multicellular organisms in wild type cells. So these pictures are to the exact same scale. What you can see is that the SMCL1, when it's at the mound stage, is much bigger than the wild type mounds. And similarly, at the fruiting body stage, it's producing really this massive fruiting body, which the wild type cells were not. So when Simon quantified this, he found that there was roughly a 3-fold increase in the amount of cells which were making up the multicellular organism. So in that regard, SMCL1 is sufficient for this particular trait of multicellularity. We're also interested in molecularly characterizing these genes, and we've just begun this, where we molecularly characterized HDAD, which was a putative histone deacetylase based on its homology to mammalian proteins. So what Simon found was that when he knocked out this gene, it caused an increase in H3K27 acetylation relative to the wild type. And conversely, when he overexpressed this gene, it caused a decrease in H3K27, suggesting in vivo that HDID could be an H3K27 deacetylase. However, we wanted to test this directly, so Simon and Henry GSD tagged and bacterially purified HDAD and then performed in vitro deacetylation assays with histones, which he had purified from Dictionalium. And what he found was that HDAD could indeed directly deacetylate histones and decrease the amount of H3K27 acetylation, suggesting that both in vitro and in vivo, HDAD really is a direct H3K27 deacetylase. And finally, the last piece of data, which I'll show you, Simon wanted to examine how this deacetylase was affecting chromatin structure more broadly. So what he did was he performed electron microscopy experiments, and this is just kind of zoomed in on the nucleus of the Dictionalium, where you can quantify the amount of heterochromatin, which is the more highly condensed inactive chromatin versus the euchromatin. And so here's just the wild type cells where the nucleus is outlined in yellow and the nucleolus in blue. And when he quantified the amount of heterochromatin, what he found was that upon treatment with trichostatin A, this histone acetylation inhibitor, he saw a decrease he quantified a decrease in the amount of heterochromatin. And similarly, when he knocked out HDAD, this also caused a significant decrease in the amount of heterochromatin. So this kind of brought this whole project full circle, where we had identified bioinformatically that the epigenome of at HDAD was marking it as a multicellular gene, and this is a gene which itself is affecting chromatin modifications and chromatin accessibility. So what we found in conclusion was we identified that Dictionalium does have these very distinct epigenomic profiles at the different cellular stages. We identified chromatin modifications which correlate with gene expression patterns, and they can maybe help us to identify stage-specific expression. So the chromatin modifications might help to drive stage-specific expression. Through this comparison of homologous genes, Simon was able to identify a multicellular epigenetic signature. And then furthermore, he was able to manipulate those genes, both by deletion and by overexpression, that he had identified based on their epigenetic multicellular signature, and showed that they were both necessary and sufficient for multicellularity in Dictionalium. So some of the next steps that we're currently beginning to address for this project is we want to examine whether chromatin modifications themselves are sufficient to drive multicellularity. So if we change the epigenome at particular loci, is that sufficient to induce multicellularity? We're also very interested in characterizing these 52 other genes that we identified as being important based on their multicellular epigenetic signature. We also, so this initial analysis was done based on the ATAC-seq data, the chromatin accessibility data, but we also have this chromatin modification data. And so we want to see if there are specific histone modifications, which can also be used to further refine which genes are important for multicellularity. We're currently performing single-cell ATAC-seq to try and identify whether changes in the epigenome help to determine whether a cell is predetermined to be an altruistic cell or a selfish cell. And we're also interested in examining epigenetic memory and how that can regulate, how that can be regulated in Dictionalium. So finally, in conclusion, I'd just like to thank the people who did this work. So the first project on the ribosomal RNA methylation was a very nice collaborative project within the lab, spearheaded experimentally by Noah Liberman and Zacho Brown, 2 postdocs, as well as Andrew Earle, a rotation student, and myself. And we were also helped by a number of people experimentally within the lab. And we were also helped by Maxim Gerashenko, a postdoc in Vadim Gladishev's lab, who performed the ribosome sequencing and analysis for us. For the second project, this was largely driven by a very talented postdoc, Simon Wong, who's going to be going on the job market soon. Exceptionally talented. He can do both wet and dry lab stuff. But he was also helped by a number of undergraduates and graduate students within the lab. and also helped by Elizabeth Polina, a postdoc in the Greenberg lab, for performing some of the ChIP-seq and ATAC-seq, Lindsay Pino, a postdoc in Ben Garcia's lab, who performed some quantitative mass spectrometry that I didn't present here, and by Yihao Wang, a graduate student in Paul Greer's lab, who performed some of the single-cell RNA sequencing and the subsequent analysis. So thank you for your attention, and I'd be happy to take any questions.
Thanks, that was great. I need to find my reaction too. Questions? So I actually have a few questions for the unicellular to multicellular sort of part of your talk. So you had found originally you had like the conserved class of genes, and then you had a class of multicellular genes that you had identified based on the sort of open or closed chromatin state.
Yep.
So did you see any type of functional enrichment or any kind of chromosomal enrichment of those genes?
Yeah, that's a great question. We didn't see any particular localization of all the genes in one location. They seem to be kind of randomly distributed. We were immediately kind of attracted to the genes like HDAD, which could potentially regulate chromatin, just because that's what we're interested in. There were also there were a bunch of ribosomal genes, which seemed to change, also harkening back to the initial portion of the talk, where the ribosome, there is a dramatic shift in which ribosomal proteins are being produced. But yeah, we didn't see other than what we observed based on eye, we didn't see any particular enrichment, but also the Dictionalium genome isn't as well characterized as the mammalian genome. So if you do like a gene set enrichment analysis, it's going to be kind of geared towards whatever processes are most well studied in Dictionalium. Yeah, so we're really interested in characterizing those genes further and seeing what roles they're really playing. But yeah.
Kevin, do you mind if I ask my question first, because I have to pop off very soon for next meeting, but I really enjoyed your talk, Eric. I've been polluted by reading Mike Lynch over the last year. This is the book I've been reading most assiduously.
Gotcha.
And in the last chapter of it, he goes on a rant, a total rant, I kind of enjoy it, about adaptionist approaches to everything, specifically with respect to the increase in organismal complexity and going from unicellular to metazoan. And he makes the case that everything, essentially, in his opinion, is driven by population size. And he, in matter of fact, he says that you can't understand anything in evolution without looking at population genetics and the NP. And so his take on fancy ass molecular networks is they're a total kludge driven by low n. Now, Dictionalium probably doesn't have that low n problem, but those of us who are big organisms certainly do. And a big organism is a C. elegans. That is a big organism. So the NP is not sufficient really to winnow away deleterious alleles very efficiently unless you're really a unicellular organism. So I'm just asking you to kind of speculate on that big issue.
Yeah. It's a good question. I mean, I think that, so obviously what we see, the end product, is whatever survived. So, I mean, I think that the capacity for multicellular organisms to survive, we're only seeing the end product. But basically, I think that multicellularity evolved many different times, and there's probably convergent evolution. So it's been said that they evolved at least 53 times independently, and there's sometimes when organisms drop down from multicellular to unicellular. But yeah, I think that that it's going to be a convergence of multiple factors, and I'm not saying that epigenetics is the sole driver of multicellularity. I just think that it's an important transition.
I'm totally comfortable with that. So it's not a how question, it's a why question.
Yeah.
It's driving it. And there's kind of an implication that it's all about selection. It's not. It's often for metazoans all about drift.
Yeah. I think, yeah, whatever is going to survive. I mean, so for the Dictionalium, what we see is that this is an organism. The only way that it's surviving these periods of drought or no food availability is through adapting this multicellular stage. So, it's going to be a bottleneck. Any genes which are present during that phase, which make it into that system, they're going to be the ones which are going to propagate and then get transmitted to the next generation.
I put his PNAS paper in the chat.
Yeah, thanks. Yeah, I'll definitely check it out.
Probably fascinate and horrify you.
No, it sounds like an interesting book. It's been so fun to get into this field. The EcoEvo, the theoretical papers, and they're just so beautifully written. It's such a pleasure to read them. When you're used to some, biochemistry papers, these ones, they're just so elegantly written. I really have been enjoying reading the theories.
Gotta leave. So.
Hi. So I have sort of a chicken versus egg question. So when you think about this in terms of chromatin remodeling, I sort of classically think of transcription factors leading the charge in terms of, transdifferentiation or sort of remodeling like Yamanaka factors or pioneering transcription factors. So which direction do you think it's going? Is it transcription factors bringing in the remodelers or the remodelers dictating the transition states?
I think it's probably both. I'm going to take a sitting right on the fence opinion. So HDAD, for instance, that has been shown to associate with these MADS-Box transcription factors in other organisms. So I think that there are going to be some instances where the chromatin reorganization is going to proceed the transcription factor's capacity to sit down. There are other instances where you're going to have pioneering factors which will sit down and cause the chromatin to become open and accessible. And sometimes those two pathways are molecularly directly interacting, as is the case with HDAD and these MADS-Box transcription factors. In some cases, I think they're going to be independent of each other. So yeah, I think that probably both are. I mean, there's reasons. There's experimental reasons to think that both are in play, I think. But yeah.
Yeah, and then in terms of the sort of who becomes what in the multicellular organism, do you believe it's stochastic or is it maybe just random positional? So if you have cells all around you, you're going to more likely become X, while if you have cells on one side, you're more likely to become Y or something of that? Or do you have a sense of that at all?
Yeah, I think that's a great question. I think we now have the tools that we can maybe begin to address that question experimentally. So I think with some of the single cell techniques, we can first quantify. So our idea for the next steps is to first, quantify it, see what kind of diversity there is. And I'm sure there's going to be diversity within the single cells at the vegetative state. But then also we can do some of these like merfish and saberfish, where you can visualize specific transcription factors within cells. And also what I want to do is maybe sort those cells out, fact sort them based on their properties, maybe what we think epigenetic factors,
Right.
and fact sort them out and then see if you put them into a naive population, do they drive it, or are they just kind of passengers? Are they the first cells to become multicellular? So I don't know the answer to the question. I mean, I think that there is this natural stochasticity and there is periodicity in Dictionalium, like there is in most organisms. So I think probably it'll be a combination of both, where some cells are going to be, more prone to become the leading edge of the Dictionalium slug because they're in a particular stage of expression which predestines them to it. But I also think that there's going to be some epigenetic component where some at least this is my theory, where some subpopulation of the cells are more likely to become, say, the altruistic cells, whereas other cells are more likely to maintain their selfish nature. But I think we're just poised to be able to address that question. I think it's a really fascinating question.
Yeah, I mean, it's a classic question throughout development and sort of everywhere. So yeah, it's a fascinating organism. I think it's a genius to use it, to be honest. I think it's a good idea.
Thank you. Thank you. Yeah, we're very excited about it. I think it's got a lot of potential for addressing some really exciting questions.
So there is a question in the chat. If methylation on ribosome by methyl 5 is a reversible reaction, are there any known mechanisms, reactions, or conditions for demethylation? That's
a great question. So for methyl 5, it seems to be a mostly constitutive methylation event. So we see it occurring in 98% of the time. Surprisingly, when we stress the worms, we don't really see changes in the methylation event, at least at the level that we're quantifying it at. And we haven't yet identified a 18S adenosine 1717 demethylase. That doesn't mean that it doesn't exist, but it might be that it's just kind of the turnover of the ribosome, which eventually leads to its elimination. There are other methylation events in the ribosomal RNA which are substoichiometric, which aren't occurring in 100% of the ribosomes. And I think those ones are more likely to be in that tunable on-off stage and more likely to actually be important for maybe ribosome heterogeneity. And we have some other projects studying some other ones of those, but they're a little too preliminary to talk about here.
Yeah, so that's interesting. Maybe you could speak to sort of, if this is a sort of constitutive mark and process, but it decreases fitness under stress conditions, like what's going on?
Yeah, that's definitely been puzzling us, and it kind of broke our hypothesis a little bit. Yeah, I don't know. I mean, I think that it's probably stress in combination with the translation of this gene, and its specialized translation. So I think it's a confluence of multiple factors. But yeah, also why the organism would evolve to have this modification, which is making it less fit. We don't know that. It's probably some instances of antagonistic pleiotropy where it's good for one particular stress, but not for something else which we couldn't quantify. Everything which we measured, it seemed to either be beneficial deleting this modification or have no effect. So I don't know why this would be conserved and selected for, but it is well conserved. This modification is present in many different organisms. So yeah, it must be under some conditions that we haven't yet examined.
Oops. Yeah, sorry. Yeah, so the phone call, it doesn't die. Yeah, so I was thinking, so there was conservation of that position that was methylated in humans and in worms. Is there genetic variation of that position, or is that invariant?
That residue is pretty well conserved in the ribosome, and it exists kind of at the interface between the ribosome and the messenger RNA. It's at that recognition site. So that might help to provide some of the specificity as to why methyl 5 is recognizing CYP29, why this 18S methylation is important for binding to CYP29A3. But yeah, the ribosome in general, a lot of these methylation sites, they are well conserved from yeast to humans. Sometimes if there's a lack of methylation in one instance, it'll be added in another place. The ribosome RNA is slightly different sizes. It's gotten bigger and bigger as we've gone up in evolution. So that might also help to confer some of the increased level of control that the ribosome might play in more recently evolved eukaryotes. But yeah, in terms of this specific site, it does seem to be well conserved, and the methylation seems to be well conserved.
And so the metal 5, have you found any other targets, or
yes, so far not. We haven't, we looked at messenger RNA methylation to see if there was any change. We didn't identify any, but we just looked by global assessment. So we looked at by HPLC. I think to do it in more detail, it would require doing some sort of specialized sequencing technique. But we didn't identify any other substrates. The ribosome and the ribosomal RNA is a very prevalent, species of RNA. So ribosomal RNA constitutes 80 to 90% of total RNA. So, and there in C. elegans, there are 54 copies of the 18S. So it has a big job methylating just that site. But yeah, we weren't able to identify any other methylation events, but that doesn't mean that they don't exist. Particularly maybe within that motif, which is present in the 18S, but it might also require the three-dimensional folding for that motif to really become the right motif. But yeah, we'd have to examine it in more detail. Maybe the best way to do it would be to perform RIP sequencing to immunoprecipitate any RNAs bound to metal 5 and sequence them. But we haven't yet done that.
Yeah, because I think in the paper you gave to the graduate students, you were talking about how sort of this mechanism may be conserved of metal 5, methylating 18S, but there might be other cofactors involved in, like, not cofactors, but they may need other partners to do this in humans.
Yeah. There's a group, the LaFontaine group, which identified that the mammalian metal 5 had this necessary cofactor, which was required for its capacity to methylate the 18S. We tested that same protein, the homologue of that protein in C. elegans, and it didn't have any role in methylating, both in vitro and in vivo, in methylating the 18S at this residue. So yeah, there might be more complexity which evolved upon the transition to humans. But yeah.
And that can't be described by maybe, so I don't know how conserved the metal 5 is between the C. elegans and humans. Could it have something to do with the metal 5 having, I don't know, additional property? I don't know how that conservation.
Yeah, well, so oftentimes a lot of these epigenetic modifying enzymes, they exist in multi-protein complexes which help to confer their specificity. So it's why, like, an H3K4 trimethylation complex doesn't methylate every single H3K4 H3K4 in the entire genome. It methylates specific ones, and that's because it's bound to different proteins associated with unique RNAs, which help to direct it to its specific targets. The same thing is probably in play for these RNA methyltransferases as well. I think that a lot of the, it'll be interesting also to examine where metal 5 is functioning within the cell. A lot of these ribosomal RNA modifications occur in the nucleolus when there's this processing of the ribosomal RNA. So that also might be a part of the reason why it's specified to a unique transcript, but we haven't yet examined that specificity by localization in C. elegans.
Cool, thank you. Sorry for all the questions. Any other questions from the audience, graduate students?
I have another question going back to the Dictionalium. I'm obviously fascinated by it. So have you thought, or are you, is Quorum Sensing something that's known to be involved in this process? Because obviously unicellular organisms can use that to communicate. Is there something equivalent that's existing in this organism?
There must be, because the Dictionalium always aggregate to the same size for their the multicellular organism. Well, I mean, it's always around the same size. It's 215,000 cells on average. making the multicellular organism. So, but it's always the same size, and you have many of these multicellular organisms in a dish. So somehow they must be sensing that. But I frankly don't know enough about it because I'm but it would be worth looking at, especially how that how that signaling plays into on a mechanistic level, how it plays into the multicellularity. But I just don't I don't know the answer to it.
Right, because the other interesting thing would be, is there signaling gradients, something that basically establishes a gradient to help.
Yeah. So for that, they identified that cyclic AMP is the secreted molecule, and they do aggregate towards it. So if you spot a bit of cyclic AMP, you'll see them kind of moving towards it. It's really fun to watch. And a lot of the work in the in the 70s and 80s on Dictionalium did identify cyclic AMP as that signaling molecule, and identify the signaling pathway which it elicited in each of the cells.
You could race Dictionalium and have bets on it by how fast they can go across.
Yeah. It would be cool to have like GFP expressing Dictionalium and not GFP. So, yeah.
If you want to start a company with me on betting for Dictionalium, we can talk later. But
that sounds good.
Thank you very much.
Yeah, my pleasure.
Yeah, so the Dictionalium are really interesting. I know you had mentioned that like multicellularity had evolved multiple times. So like where do Dictio fit into that phylogeny? Like are they sort of recent, later? Like why, and is it rare for organisms to employ both? I'm assuming that it's rare.
Yeah. So it's exceptionally rare. I mean, a lot of the organisms within the Dictyosteliae clade do this, have both unicellular and multicellular, but outside of that, there are very few organisms which are both. Nicole King studies this dinoflagellate, which aggregates. There are 5 of them which aggregate to form a multicellular organism, and she also does some really cool work studying multicellularity in that. In terms of where on the evolutionary scale, I mean, I see the phylograms that people draw, but I never really know for sure how they got there, and how you can have any confidence in something that happened 900 million years ago. I mean, I think it's all due to comparisons of the genome and how it's evolved. I understand that, but but that's my ignorance. I'm sure they know what they're doing. So yeah, it's Dictyostelia, it's this algae. It's kind of right around the same place as S. cerevisiae, but again, it's still 1,400 million years separated from S. cerevisiae, and 1,400 million years separated from C. elegans. So it's a very wide range. I don't think that people who know what they're talking about, these who do phylograms, put Dictyostelium actually as the transition point of the last known common unicellular ancestor. I don't think they place it there. So I think it evolved later. But as I said, it's really fascinating to think of, because there are some of these organisms which have dropped down from multicellular to become unicellular, because it's more environmentally, it's more fit for them to have evolved that way. So I think those questions are fascinating. I just don't know enough to really be able to answer them in a intelligent and
yeah, no worries. I was just curious, and then sort of final random question out of curiosity. So is there something about the environment of Dictyo that has caused them to adapt this? Is it a particularly harsh environment that they live in?
Yeah, no, it's a soil living amoeba. I don't think there's anything particularly harsh, but they, in their critical evolutionary period, they must have been in a phase where food availability would be the limiting factor. C. elegans also, when they starve out a plate, they enter this kind of dour phase, which allows them to persist without food. So I think organisms naturally adapt to their environment, and it must have happened when Dictyostelium was evolving that it only the ones which could survive this period of food scarcity would be able to propagate. And they did that by transitioning to this multicellular stage and then sending their spores further away to allow them to find food. So only the ones which could maintain their population at the appropriate density would persist and propagate themselves. But yeah, it's a really interesting question to think about. I'm sorry I don't have a good experimental answer to it.
Any last questions? I mean, I think it's such a fascinating model. So yeah, a lot of these are kind of like open questions.
That's part of what drew me to this organism, is there's so much cool stuff out there. And you know, there are a lot of Dictionalium labs who you know, they are predominantly studying this chemotaxis capacity, but there's a lot of cool stuff out there, and I need to immerse myself more in their literature and figure out more about what's going on there. But yeah, we're really, I mean, as I said, we're really attracted to these questions about epigenetics and how it's controlling these critical processes like multicellularity, like cellular altruism, and we think it's really powerful for addressing some of those questions.
Indeed, yeah. Fascinating talk, fascinating topics.
Thank you, thank you.
Any last questions, grad students? Now you've had a lot to think about. Going once, twice. Well, I guess if there's no more questions, thank you for accepting BJ's invitation. It's really great, fascinating stuff. Yeah, really exciting.
It's my pleasure. I enjoyed it. Thanks for having me.
Well, thanks everybody, and thank you, Eric, and have a great weekend.
Yes.